Last time we covered parasitic roundworms, the nematodes, and today we want to talk about parasitic flatworms. When people think of flatworms, they often think of tapeworms, and this is what this artist rendition is showing here. It's a intestinal wall, and you can see the tapeworm that's there living in the intestine. It's got these segments called proglottids, which we're going to talk about. But there's actually two different classes that we want to talk about that are both different types of flatworms. So there are the cestodes, which include the tapeworms that I was just describing, and they look like tape. They're very flat. In fact, this is another artist rendition, but it doesn't look, it's very flat. It almost doesn't show very well how really thin the thickness of these organisms is. And then there are the trematodes, and those include flukes and organisms like schistosoma and Clenorchis. There are many different tapeworms, and Tinea is a good representative example. This is actually a photograph of Tinea, and you can see how flat it is, and you can really get a sense of why they call it a tapeworm. But their anatomy is fairly straightforward. They have a scolex. It talks about that being a head, but that's really called a scolex. This is a picture under the microscope of a Tinea scolex. And this attaches to the intestine of the definitive host. And so the definitive host is where the adult worms live and reproduce. So in the case of the pork tapeworm, you would be the definitive host because you're the host that they reproduce in. If you look further down the head, you see these little segments beginning to show up. These are called proglottids, and they actually are continuously produced by the neck. And as new ones are produced, the old ones end up being pushed further and further down, and they mature. They contain eggs, testes and ovaries, eggs and sperm. And as the proglottids get older, they mature into gravid proglottids containing fertilized eggs that are ready to form new worms, and eventually these proglottids actually fall off. And so the adult worm is still in the intestine, but it's releasing its eggs in these little mature segments called proglottids. If you've ever had a cat with fleas or a dog with fleas, you might have seen where the animal sleeps. They look like little grains of rice, and sometimes they're moving. And so that's an indication that your pet has tapeworms. And that also means your pet has fleas, because fleas are an intermediate host for the tapeworms that infect dogs and cats. Tapeworms are hermaphrodites, so there's no separate male and female. They self-fertilize, and very convenient. Another thing about tapeworms that's interesting is because they're so flat, they don't need a digestive system. So they just absorb nutrients from their host, and they absorb those nutrients through their cuticle, which is the outer layer. People have thought that maybe if they just went ahead and ingested a tapeworm, they could, eat more food without gaining weight, because the tapeworm, you'd have to feed yourself and the tapeworm. But unfortunately, tapeworms don't require a lot of nutrition, and there are other problems with having a tapeworm that we're going to talk about. So this is the tapeworm life cycle here. Let's start with a pig tapeworm, the Tinea solium. So here's our little piggy. And this little piggy ate some tapeworm eggs, and those eggs hatched. They penetrated the intestinal wall of the pig, and they ended up encysting in the muscle of the pig. And so these cysts are called cystoceri. And let's say you are eating this pig, and when you cooked it, you did not use a meat thermometer and ensure that your meat got to an internal cooking temperature of at least 145 degrees Fahrenheit. And if this happens, then you may ingest a live cystoceri, and when they get into you, what'll happen is they will excyst. They'll come out of their cyst, and they'll mature into an adult tapeworm that attaches using its scolex to the intestine. And as it attaches to the intestine, it's going to then begin to grow and produce these proglottids, which will eventually fall off full of fertilized eggs, and they'll be released in your feces. Now, that doesn't sound terrible, and it's not, as long as you never ingest those eggs. Because if you ingest the eggs, you actually end up in a completely different place than if you ingest the cysts. If you ingest the eggs, then those eggs are actually going to hatch into the oncospheres and penetrate your intestinal wall, and they will circulate in your body, and they will encysst in your muscle but they will also insist in other tissues, including the brain and the eyes. And so this causes a very nasty illness called cysticercosis. And that's what I'm going to talk about on my next slide. I do want to take a little detour though and just mention that dogs and cats are frequent hosts for tapeworms, particularly those that don't get treated for fleas. So here's a dog you can see that they've shown the tapeworm inside the dog here. And the tapeworm is producing those proglottids, which then will leave in the feces of the dog. They end up ingested by the fleas. The dog will then when he's getting bitten by the fleas, he'll chew on them and he'll ingest some of the fleas, and then the flea will release the further larval stage that allows that worm to develop that again in the intestine. So that's the life cycle. Very, very common for dogs and cats to have tapeworms if they also have fleas. And that's why using flea medicine on them, things like Advantage or Revolution, is actually really important to the health of your pet. Not only are fleas really nasty little ectoparasites and they can cause anemia and make animals really miserable, but they also are hosts to these tapeworms. The dog is the definitive host, and the flea is the intermediate host. So definitely don't allow your animals to have fleas. There was a time prior to the year 2000, I don't remember exactly what year Advantage first came out, but it was a constant battle living with pets with fleas. We were constantly combing the fleas off and treating them for tapeworms, and everywhere they slept, there'd be little proglottids moving around that were from the tapeworms, and they'd be in your bed if the dog slept in your bed. And we got used to it, but it was super disgusting. And now that I haven't lived with fleas for a lot of years, I think it's kind of unbelievable that we put up with that. We really love our pets. I think that's the reason. All right, I said that in a human, let's go back and just review. So we said that in a human, if you eat the pig, you get the tapeworm. But if you eat the eggs, you get these little oncospheres insisting in your muscle and in your brain. And that's what we see here. So this is actually, if you get it insisting in your muscle, it's not that big a deal. Usually there's no symptoms. Maybe you get some lumps under your skin. But if those cystoceri are in your brain, then we call that neurocysticercosis. That's the most serious form of the disease. They can also insist in your spinal cord. You can see the brain tissue here with holes in it. So this has very serious consequences. A lot of times it's associated with confusion, or somebody seems like they're developing dementia. It can cause headaches, encephalitis, ultimately stroke and death. So this is a really important reason to cook your pork 245 degrees Fahrenheit to a final internal temperature, and then wash your hands, right? That if you're doing both of those things, you should be able to avoid cysticercosis. All right, so that's class Cestota, the the tapeworms, and then we have the trematodes, which are the flukes. And the flukes have very complex life cycle. There's usually 2 hosts at least. The first intermediate host is typically a mollusk, like a snail. And then there's a definitive host, which is a vertebrate. This is where the adults will live and undergo sexual reproduction. These are flattened, but they're not quite as flat as a tapeworm. And because of that, they have a digestive system, but it's primitive compared to the nematodes. So they have a mouth that food can go in, and then on either side of the mouth, they have this ceca. It's maybe easier to see here, sort of on either side, but you see that it's blind-ended. It doesn't end in an anus. And so what happens is the food goes into the ceca, digestion occurs, and then waste products get expelled back out through the pharynx. So that's one opening for food in and food out. And it sounds really gross, but the trematodes don't seem to mind, and they're pretty gross animals anyway. They're hermaphrodites, so they have both the ovary and the testes are in the same organism. Okay, then we have a uterus here where the eggs will be fertilized. So these organisms are hermaphrodites. They're both male and female reproductive structures in the same organism. Okay, now I'm going to put this video down below for you. This is showing the life cycle of Clonorchis. So that's something for you to watch after you're done with the overview that I'm providing. But I do want to go through the life cycle of Clonorchis as well. So Clonorchis is pretty complicated. So here's how this life cycle works. So here we have let's start with the release of eggs passed in feces. And these eggs then are going to swim to a snail or get absorbed by a snail. And then in the snail, they're going to go through several larval stages that terminates in this cercariae. The cercariae is a swimming larval stage, and when it swims, it doesn't actually go back to the human. First, it insists on another host called some kind of fish. It's usually a freshwater fish, and then it insists in the mussel. And then the human, which is the definitive host, will eat the fish. If the fish isn't fully cooked, then those the metacercariae that have insisted in the fish will get into your digestive system. They will exist in the duodenum, part of your digestive system. And the adults will migrate into the biliary duct of the host, which is you. And so if we just do a quick look over here, this is the human liver, and these biliary ducts kind of extend into the liver. It's not really showing all of them, but basically what you end up is having these Clonorchus worms in your liver, and they cause inflammation of the bile ducts. They can block the bile ducts, but the problem is that you tend to be infected chronically with them, and long-term inflammation in the liver leads to a much higher risk of liver cancer. So that's the concern with Clonorchus. And again, adults who are infected will ultimately pass the eggs into their feces, and the cycle goes over and over again. The video we skipped that you're going to watch later, it actually talks a little bit about some of the challenges in trying to educate people about eating raw fish. It's very hard to get humans to change behavior, particularly around sex and food. Those are the two things that we have a lot of trouble with. And so trying to explain to people that it's dangerous to eat raw fish in this particular case, if that it's not in clean water, etc. That's a project in terms of eliminating this particular pathogen. The last flatworm we want to talk about is called Schistosoma, and there's actually 4 different species. I'm sorry, there's 5 different species, but we're just going to talk about Schistosoma mansoni. And again, this has a complex life cycle. This particular case, though, there's only one intermediate host, and that's the snail. And then the cercariae, instead of insisting in a fish like the liver fluke did, it actually insists in the human. It gets into your skin. So let's just start with the excretion of the eggs, and we'll go from there. So here's a human, and what happens with Schistosoma, depending on the species, it either lives, the adults live in the blood vessels that surround your bladder or your colon, and they produce eggs, and the eggs will go through the blood vessels into the colon and into the bladder, and you release them in either your urine or your feces, and sometimes there's some crossover between species. So some species are more associated with being passed in feces, and some tend to be passed in urine, but they can do both. So Schistosoma mansoni is typically passed in feces. Once the fertilized eggs are released, they hatch, they release the larva that then ends up in the snail. That larval stage is finished, and then we have 2 more larval stages that end in the cercariae again. So this is that free swimming larval stage, and this is going to then penetrate the skin. So if the person is pooping, like humans are pooping or peeing in water, and then they're going in the water, the larva can penetrate the skin. Once the larvae get into the skin, they get into your circulation, they migrate to the portal blood in the liver, and they mature into adults. And here's our adult Schistosoma. And again, they are going to go back to those blood vessels that surround the bladder or the bowel and release eggs that then migrate into the bladder or the bowel. Jimmy Carter and the Carter Center have been working on Schistosoma, just, almost an unknown disease that causes so much death. And they've done really admirable work to try to reduce the burden of schistosomiasis. So I'll have you watch this video. I'll place it below this initial overview that I'm doing, so you can take a look at it and hear about that. All right, so these are the stages of Schistosoma mansoni that you should be familiar with that could show up on a lab practical, for example. Let's go through the life cycle again. So here we have our human host, and remember that the inside the human, the mature male and female flukes are producing eggs, which are going to go through the intestines or the bladder. In the case of Schistosoma mansoni, it tends to be the intestines. They pass out of feces and or urine. You have that Parasitia, which goes into the snail, which then, as the larval stages progress, you have the free swimming Cercaria, and they're going to swim back into the skin and get into the host and get into the bloodstream, and it's going to start the process all over again. For Schistosoma mansoni, you should be able to recognize the egg. It's got this really cool little spike on it. So Schistosoma mansoni eggs are easy to identify. You should be able to recognize the Cercaria as well as the adults. So this is a male and a female schistosoma, and they're flat, but the male is sort of wrapped around the female. This is really unusual because remember we said that platyhelminthes generally were hermaphrodites. The male and female structures, reproductive structures, are in the same organism, but schistosoma is the exception. And you know, in class how I always say, you know, when someone says, is it always such and such, I say, never say always in biology, never say never, never say always. This is an example of why I get nervous trying to do that, because schistosoma is one of those organisms that by all accounts should be a hermaphrodite, but it's not. It has male and female. The male and female are dimorphous, and you can actually tell the difference between them. So these guys live their lives having sex, producing eggs, and generally causing a lot of ill health in humans. So we said that the parasites, as they're making eggs, right, the adults are happily procreating in the blood vessels around the colon and the bladder, and they make all these little eggs, and the eggs pass through the intestine or the bladder. Now the problem arises when some of those eggs don't make it. So they can get lodged in the intestine or bladder and cause inflammation. They can actually do damage to the spleen, the liver. Oftentimes, people with schistosomiasis have chronic schistosomiasis. They're constantly being reinfected. And over time, that's going to result in damage to the liver. So you see children like this with an enlarged liver from chronic schistosomiasis. Ultimately, you know, people who live with this for many years end up with increased risk of cancer as well because of the chronic inflammation. So about 240 million people worldwide. Most of the burden is in Africa. And just knowing that it's second only to malaria in terms of the number of infections. And it's amazing how few people have heard of schistosomiasis given how many people in the world suffer from it. This is a great article. The link is active in the slides, so you can have a peek to it if you're interested in it. But this was in 2018. This was published. They're actually trying to develop a vaccine for schistosomiasis. And they took some healthy volunteers in the Netherlands, and what they did was they paid them $1,200 each, which I don't think is enough. And they infected them with only male worms. So the idea here is that they couldn't be hurt by these worms because they can't make eggs, right? They can't fertilize eggs that leave. And they're doing this to explore the possibility of a vaccine. So they're basically giving them a vaccine and then infecting them deliberately and looking to see how effective the vaccine is. And they know they can give them praziquantel, which is a drug that is very effective against schistosomiasis, and it's very inexpensive too. You'll hear about it when you watch that video that the Carter Center produced about the work they do against the work they do to try to help eradicate schistosomiasis. Anyway, it's an interesting article, and I love in particular this picture because you can see the male worm is in brown and the female worm is in green, and you can kind of see how he's wrapped around her, cuddling her for their whole life. What a good guy. All right, my last video is a TED Talk from 2013, which seems old now, but it's still one of the very best TED Talks I've ever seen. Ed Yong, who is the author of I Contain Multitudes, he's really an expert in the microbiome, talks about a variety of different parasites, their ubiquity on Earth, their contribution to the ecosystem, and how many parasites seem to be able to change the behavior of their hosts. And that doesn't mean other animals, it also means humans. So I want you to watch this and pay attention to it and take some notes. And to encourage you to do that, I'm going to make sure that some of the information here makes its way to the lab practical. So hopefully that's a motivation for you, because I don't want you to miss it. This is one of the very best TED Talks I've ever seen. All right, if you have questions, I'm always available by email, or you can also join my online office hours. Hope to see you soon.